with a keynote lecture given by Ge Gao, who is principal investigator at the Peking University, but he's also a Chinese or board member of the Chinese Society of Biotechnology and of Bioinformatics. And he will talk to us about towards a rational designed regulatory language model. Welcome, and it's a pleasure.
Yeah, thanks so much for the chair and committee for this great opportunity. Actually, as I was just talking about that, it's really nice to meet you all of those in person. I still remember the last time I met with Sarah in person, before the pandemic, the BEP. Yeah, so, I was trained as a pure mathematician before jumping into the bioinformatics and genomics. So it's really nice to have the great opportunity to present our recent work on modeling the cell regulatory map. So we humans are essentially an assembly of trillions of cells. Most of those cells share a common genome, but different expression and distinct functions. And the gene expression is a highly regulated process, which involves the genetics and epigenetics regulation layers and the sophisticated interaction between and within those layers. So since the discovery of the transfer factor 60 or probably 70 years ago, we now have known that the gene expression is a nonlinear, hierarchical, and combinatorial process. So our idea is to model such a complex system through generative models or computationally. We will try to have the full distribution of the input data that we actually like to have the PXY, the full data distribution. Not only, things we are interested or like the signal, but also things we are, generally not interested like the noises. So as you may see already, such task is intrinsically hard because, in reality, we usually do not and actually cannot have the, the full data set as input. So we just need another, assumption incorporated that there is a low-dimensional root site behind, the very complex, high-dimensional observation. The root site by itself could not be observed directly, but we can make our model learn from the observation, as it is exactly the root site that generated those observations. So just like what the ChatGPT does, right? The ChatGPT does not keep all inputs as is, right? But tries to learn, the underlying rules of human speaking language, so that it can simulate and understand what we say and respond accordingly. So similar to ChatGPT, we are here going to learn the language of the cell regulation, or the cell regulatory language. We are trying to build a cell regulatory language model. But different from the ChatGPT, the basic unit of our cell regulatory language model is not word or sentence, but the gene, or more precisely, the expression and the regulation of genes, and the gene module, the pathway, or things like this. But one thing to note here is that the success of the disk-driven approach used by ChatGPT roots from the fact that it's rather easy to generate and verify the human speaking language, right? So that's the scaling law. More computational power, larger model size, more parameters, and the larger this size, and you just magically get some more powerful model, right? But let's say it's not true for our cell regulatory language model, because the generation and the validation of the our cell regulatory language just need extensively experiment that's both costful and time consuming. So we just need another component that is a knowledge part to make sure that our model could be more cost effective and cost efficient during not only the training but also the prediction or the inference. So let's back to the the BP, before pandemic, the 2017, when we begin from the single cell RNA-seq. As what I mentioned before, we basically model the observed data X, expression data X, as being generated from the lower dimensional latent space U, by a particular transformation presented as an encoder or decoder or some computational jigs just coded as an autoencoder, encoder decoder neural network structures. And specifically, we also incorporate a dedicated adversary component to model the systematical differences across different inputted samples. Part of those differences are usually coded as a batch. So we can see the model can handle batch effectively, as the pancreatic disk site shown. The model is able to align cells of the same type from the distinct disk sites, which even those original disk sites show strong batch effects. And the further benchmarks, the standard benchmarks also demonstrate that the model shown the best overview integration performance and the best resolution over several state vars at that time. So with this in the mind, we further extend our model to multi-omics. So we actually have the glue. Essentially, we introduce a new component in addition to the encoder-decoder structure. We actually have a new graph autoencoder. For the regulatory graph, we introduced as a central part of the multi-omics integration and regulatory inferences. So you can see the regulatory graph is sort of like a representation of the regulatory relationship across the genes and the promoter, the DNA methylation region of those regulatory elements across the genome. So from the perspective of the cells, the intermodality feature embedding from the graph autoencoder or in the regulatory graph could serve as a unified coordination system so that they can be used to help we integrate those data from different modalities. But on the other hand, the correlation structure in the aligned data could also be used for regulatory inferences. So mathematically, in addition to the generative architecture we used before, we incorporate a graph component to model the interactions across the regulatory layers, each with a distinct modality. In other words, the graph by itself is used for both as a regularization or a unified coordination system, and a notebook for integrative regulatory inferences. And so the model, the glue, works very well for regular to omics integration benchmarking, right? But that's something you may expect already, because if it's not, the paper would just be rejected. Okay, so yeah. But the real power of glue and its generative graph oriented design is it can do something different. For example, right, we actually have the 3 different designs across the different modalities, the RNA-seq, the attack, and the mesylome. All those data are generated by 3 different lives at the 3 different years, and our glue can automatically integrate them all together unsupervisedly. And what I mentioned before, the incorporation of the graph component not only provides the necessary guidance, right, for multi-omics alignment, but also serve as a basis for regulatory inferences. So as you can see from this, it's TF transfer factor and target network inferred by glue from a PBMC data directly. So you can see that the glue algorithm successfully pinpoint several important transfer factors like the PAX5, the EBF1, and a lot of things from the data directly. And the model can be also easily extended. For example, by splitting up the embeddings of each cells into distinct modality-specific subspecies and introduce the cross-encoder, we can easily benefit from existing simultaneous multi-omics data, such as 10x multi-omics and size-seq. So we just have the clue. So you remember the C for crossing, right? And actually, the model just won the all best for the modality matching task in the very first Europe's AI conferences for their single cell data integration challenge. And as many people already talked about this last day, we humans are multicellular organs, right? And for the cells in multicellular organs, a cell has to work with others. So for comprehensive regulatory map, we also need to incorporate the spatial context of cells. That's why we actually have the slide. Here, you can see the familiar graph, right? So each cell and its neighbors just presented as nodes, and the neighborhood relationship are just the connected edges. And also the familiar adversary component, which tries to deal with differences in this size. The resolution, the feature space, the RNA attack or mesylome or things like this, and the artifacts introduced during the experimental and probably also the data generation. So again, we can see that in the standard benchmarks, right? We have 3 representative this size from the 10x to Morphish and StrowSeq, and which cover a wide range of the throughputs and the resolution. Well, all tools works reasonably well for 10x. The slide achieves the overall best performance across all technologies. But the real power of slide, again, is its unique ability to work on the heterogeneous data, right? For example, you can see we actually have the 2 different designs for RNA-seq, spatial RNA-seq, and attack-seq, spatial attack-seq. It came with produced by different labs and with a different resolution. That's a 20 versus a 0.2, right? Some big differences. But slide can still, successfully align them unsupervisedly, and through joint regulatory inferences we just, talked a few minutes ago, we can identify some important transcription factors for heart development, right? Because all those decide about the heart. And slide can do this over more than 2 slides. Actually, by applying slide to slides at 8-10 points across the early mice development, we can reconstruct the spatial temporal development of the early mice development for many important organs precisely. And different from the intracellular data integration, right? The spatial omics comes intrinsically with 2 parts: the intracellular parts and the extracellular parts. So there's also sophisticated cross-talking between all those 2 parts. So inspired by our previous work clue, you still remember the C for crossing, right? We designed a contrastive learning framework to disentangle the cell from its context. Why? Because, specifically, we like to have those 2 related but distinct parts as, the different representation, so that each one will be more precise and, more effective and efficient. So, on the other word, we want to have a mathematical representation for the cell's inner world and the outer world. So our new algorithm, the decipher, the disentangle operation implied by the decipher just works pretty well for the standard benchmarks again. But such disentangle, such a new algorithm also enable the global analysis for hundreds of slides and millions of cells, global analysis directly. You do not need to have a different parts assembled together. You just run one algorithm and get everything well aligned, well presented. So based on those new algorithms, we greatly expanded our original HCA database we published at 2020. Now we actually have the ESA created this size for 275 million cells in human and mice across multi-modalities. And actually, we not only have those data, but also have the metadata or the phenotypic information resolved. So we actually can have the model, the AI, work without our human involved. So that's sort of like things we try to be the AI friendly, and yeah, that's something we want to. This works not only for our great biology community, but also probably for our computational guys like me. So now we actually have a set of powerful integration tools, right, for multi-omics, single cell spatial data, and large curated this size for cellular transcription, multi-species, multi-modality. But are those enough for modeling the whole regulatory system? We actually know the answer is no, because we already know from our biological study during the past 70 years that the trans part is only half the story. We also need to model the cis part, or the sequence part, right? So the idea is not entirely new, and you can see even back to 23 years ago, it has been proposed that we can take the genomic sequence as a sort of language, right? So it's a very intuitive and rather straightforward. But we'd argue that we believe that the sequence itself is more than that. Actually, we believe the genomic sequence should be taken as a superimpose of multiple languages. Say you are, a PAL2. All you are interested is, Tata, right? Tata box, and nothing else. But on the other hand, say you are a DNMT, you are enzyme for the DNA methylation. You won't bother about the Tata because there is no C and G, right? You just would focus on the CPG. But if you are enzyme for DNA replication, you may focus on the OC, right? The replication original point or things like this. So as a proof of concept, we have the SWIN. So instead of taking a holistic network as the current very popular DNA language model like the Evo2 or things like this, the SWIN uses a hybrid architecture, which include multiple different scale networks. Each will try to learn a particular language, such as language of PAL2, the language of DNMT, okay, the language of histone modification, the histone modification enzymes, and the language of the DNA replication enzymes. And yeah, as I expected, okay, the model, the SWIN, shows a superior performance than the canonical holistic network-based models. And the SWIN can also work across different conditions, like for different regulatory signals, DNA accessibility, histone modification, for different organs and tissues, and for different male-female, right? But most importantly, we like to say that the rules learned by those multi-scale networks could be applied to novel scenarios, like here for hundreds of BP's deletion as an upstream of upstream promoter of a gene. A reasonable guess, right, is that such a large-scale genetic change would lead to a decreased transcription, right? Because you just have deletion, a large-scale deletion. But the SWIN correctly inferred the opposite outcome. Actually, such deletion would instead, not decrease but increase the transcription by resharpening the local epigenetics landscape in a way that favors genetic activation. So we further extend our idea to a fully functional genomic regulatory language model, or the DNA lingo. So compared with its big guys peers, the DNA lingo is not a LLM or large language model because it's rather smaller. So it just has 80 millions of parameters, other than billions of parameters like the Evo2. However, for multiple regulatory-oriented benchmarks across not only the chromatin profiles, the transcription regulation and the post-transcription regulation, we can see that our DNA lingo just show a comparable and sometimes rather superior performance when comparing with those billions of LLMs, large language models, which confirmed our previous mindset that with a well-designed model architecture, a model can be rather cost-effective and cost-efficient than you're just following the intuitive and straightforward approach. You just use those canonical strategy. And by combining all those together, we are also working on a regulatory language foundation model. As everybody is just talking about the foundation model, right? But we named our foundation model as Wanhu, which is believed to be the first person who tries to reach the sky by rockets. And specifically, a key component of our regulatory foundation model is a causality-aware algorithm for regulatory inferences. Why we need causality? Because if we try to do something more than observation, more than correlation, we need something to understand how can we design our manipulation for a specific outcome. We need causality, right? So briefly, we model the causal dependency graph between the genes genome-widely through the masking, so that the model could be trained end-to-end through a standard backpropagation, SGD, or those deep learning tricks, which make it not only adaptive but also scalable for large heterogeneous data set. Or more intuitively, we try to find the best topological order for those genes genome-widely, which can explain the observed expression profiles and maximize the likelihood by combining the data-oriented searching and knowledge-oriented purging. Here we mean the knowledge-oriented because we will try to incorporate those things we introduced before, like the counting the chip-seq. So it's sort of like a directional edges from the transcript factor to downstream targets. And such topological sorted nature also enable a causality-aware prediction for any intervention, because we actually can travel, right? We actually can travel across the inferred causal graph and deduce the expression of each gene after the intervention, one by one, step by step, in a causality consistent approach. So you can see that the evaluation just shown that our algorithm, the cascade, just shows a rather good performance over other, the correlation or association-based algorithms. But again, the real power of the causal aware approach used by our cascade just makes the prediction more realistic and understandable, because we actually have a direct causality graph, so we can attribute the predicted change after the intervention as direct effects. Say that's knockout for the RBP9 ribosomal protein. So the direct effect is ribosomal downregulation, and the latent effect, because such dramatic change to the translation machines would lead to the cell dies. So that's all could be seen from our causal graph. right? So, but as what I mentioned just before, I believe that the real thing for our regulatory foundation model is not to interpret, but to design, right? Because if we understand something, we should have the ability to design approach to achieve any outcome we expected, right? So that's not so hard, right? Because if we actually have a reliable causality inference algorithm and a reasonable well-represented this size across all cells in our human body, right? So the design problem could be rather straightforwardly modeled as a pathfinding problem. So you actually have a sort of cells here and there. The only difference is expression. So they actually have the different positions in the cell regulatory space or cell expression space. So the things for programming or reprogramming could be rewritten as pathfinding. You just try to find the minimized operations to change the expression vectors from this to that, as long as you actually have a reasonable, precise causality algorithm, right? So as a proof of concept, we have successfully rediscovered the Yamanaka factors, or the combination of the 4 transfer factors, including the OCT4, SOX2, the KRF4, and C-mix. All those factors can effectively combine together, reprogramming the mature fibroblast cells back to pluripotent stem cell or iPS cells, and this just won the Dr. Yamanaka the Nobel Prize at 2013, right? So as you can see, our design, including not only the all 4 original Yamanaka factors, but also a cofactor that's FOXIL2, right? Which has been shown to be helpful for reprogramming by follow-up studies. So the next step is to do something more practically useful, right? So basically, we designed a combo recipes for programming to red blood cells. I'm sorry for the F1, F2, and F3, because our colleagues still want to wait the the paper was formally accepted or published. But I like to say that you can see if we actually have the transfer factors, if we have a circle and a box for the non-transfer factor. So we can see our 3 factors actually include not only the transfer factor, but also the non-transfer factor, which is usually not taken as a expectation, right? Because from the great works inspired by Yamanaka, many people just believe that the transfer factor is good enough for self-fate programming, reprogramming, or anything like this. But because our cascade, or the causality graph inferred by our algorithm would take all those genes into account. So we may actually find something different, beyond the common wisdom. And actually, we also have a large list of our design recipes, for not only the red blood cells, the hemoglobin, but also for a lot of other, cell types. You can see that the cascade design made something like the TF Atlas, which is from the great work by Zhang Feng, which doing this by the very wonderful works, the combinatorial experiments. But as we do not have so much resources for the experimental, but for the experimental based design, we try to do this in the computer. So we actually could have a wide range of choice for our target and our factors. So we actually are still working with our colleagues to verify the list, and we are also happy to have more peers join to help us see whether our idea, the causality, and the foundation model works or not. So given I actually have 35 minutes, I believe I have to conclude our works by this slide. So during the past years, we try to put our focus on the cell regulatory language model as an approach to a cell in silico, which we would define as a computational model to help us transform from the observation to targeted manipulation or design in cell, and more precisely cell fate programming and reprogramming. So actually, those work was done by a lot of peoples. Some may have actually have left my lab already, but I still like to highlight for someone still here, such as Xia Chenrui, who is a guy working on our spatial omic things like the slide and decipher. Chenrui, right there. Yeah. I believe he actually has a poster, but I don't know. I'm not sure whether the poster is has been posted, right? But he's here, and also the Ding Yang, who is in charge of the HCA, the EACA things, the curated sites, and also the Cao Zhijie, who we wish could be joined, but he's not here due to some sort of a visa problem. I'm not sure whether he's online or not. Yeah, but anyway, I'd like to thanks for the great opportunity, and also believe it's time for take questions. Thank you.
Thanks so much for the presentation and for providing such important resources to the community, and for sharing so much unpublished data as well. So questions to the speaker.
Thanks for the amazing talk. Yeah, really, really fantastic. Kind of going back to the beginning of your talk with the glue and clue sort of multi-omic integration, I was wondering whether you can give us any feeling for the accuracy of the glue, kind of the unmatched integration versus the clue. So if you have the clue, you have kind of the benchmark, and so I was just wondering how good the integration in separate kind of high-dimensional space, with the separate modalities is, versus if you have matched data and then you're integrating those data sets.
Yeah. So can you put it on screen? Actually, it's here, but
I mean, you can
not
just,
there.
maybe you can just summarize for us.
Okay.
Like how accurate is the, the unmatched, and what does it depend on, the accuracy?
Okay. So, could you put, yeah, that's terrific. So that's a terrific question. And, the clue, remember the C for crossing, right?
Yeah.
So it's sort of a different revision of the glue, because the glue is designed to work unsupervisedly for unpaired data,
Yeah.
right?
Yeah.
The clue by its design is for the, working as a semi-supervised,
Yeah.
right? So actually, according to our benchmark, that is the clue would, outperform the unsupervised glue.
But how much?
Yeah,
By how much?
At least 20%.
Okay.
Yeah.
Okay.
So that sort of, benefits from the crossing.
Okay.
Yeah. That's
So
also
it's substantial.
yeah.
Yeah.
It's a big thing.
Thank
right?
you. Thank you.
More questions to the speaker?
Thank you. That was absolutely fantastic. It's a little bit more looking at the future in terms of, you're creating this regulatory language model and then in silico cell. In a more tangible way, where do you expect to be in the next 2 years? I mean, would you be making a lot more of the predictions, or would you also be trying to reproduce them in the lab? And what would your, immediate directions of travel or areas of focus be?
Okay, I believe that's something we try to make it real, make it more realistic. But at least in my opinion, the current challenges for the ChatGPT similar language model for the for the cell probably root from two different parts. One for the model itself, because actually I don't believe the canonical model structure like the BERT, like the GPT used by the human speaking language could be directly transferred to our cell regulatory language model, because the cell is rather different. We can have the natural order for human speaking language, like we speak from the beginning to the end. But what's the order for the gene expression? It's sort of rather not straightforward, and we try to define this by causality. But that also depends on the correctness of our causality inference algorithm, right? So it's a sort of part, the model part, the computational part. But there's also another part for the data, because we human are assembly of trillions of cells, but we currently have only millions of maybe better for billions of cells in our database. So that's a big gap for the representation of our current data. So I would love to have a ChatGPT-like for our regulatory language model. I mean cell regulatory language model. But I don't think it would be an easy task, because we still need more innovation on the methodology part and accumulation on the data part. But I believe we are approaching to this. At least now we actually can have a causality based design for, I believe, light. And also, we are accumulating more data and more, resources from our great biological peers. Yeah, so I believe, we are working and we will work on this, but probably not now.
More questions? I would have a follow-up question because in the end, often looking at regulatory events and kind of signatures or gene expression signatures related to kind of specific kind of lineage commitments, how could you go back and really find these key regulators across the many systems you were looking at?
Yeah, so our current algorithm, the cascade, is designed to have, let's say, it's sort of like this one. It tries to have the computation model or computation interpretation for our observation with maximize likelihood. It's absolutely not 100% because there are still a lot of things we don't know yet. Because we actually have much lesser data for the epigenetics. We have abundant RNA-seq, rather less tag-seq, and even lesser for the mesosome, and even lesser and lesser for the histone modification. So it's sort of like the data representation problem I just mentioned. But the model by itself will try to do its best based on the current available data and resources. So you are absolutely correct that we can't do this 100% accurate, but I believe that's something we try to do at our best right now. Yeah. So that's exactly why we like to propose our list of recipes for cell programming, reprogramming, so that our dear colleagues may like to try to help us to show it's right or wrong. Yeah. I was trained as a pure mathematician, so I can't do this, but I believe that many of dear colleagues here probably may be interested in this.
Yeah, because that's obviously one of the most important future applications of a virtual cell, right? More questions? Oh, yeah.
Thanks for the very interesting talk. I had a question about the causality model, especially regarding the prior. Have you tested how the different priors improve the models, and how do you draw these edges, whether it's activated and repressive?
So you mean the repressor, right?
The CAC and PPI priors, because in that one of the last slides you show all of this network which you infer the causality on.
Excuse me, but I believe I'm not sure I fully capture your idea. So, but I mean, we currently design our model, like this. So here we define the causality for manipulation, because we are not trying to claim that we actually know every step of our regulatory circuits, but we try to distinct this from that. For the left part, we can operate on A and change the B, but not for the right one. So we try to identify, operational approach for design manipulation. That means we actually have to model the the regulation relationship across genes. So we actually do this by masking, and so we can have the things for activation, for repression. All those came as different ages, like what we have done in the glue. And so we try to have those causal dependency graph more accurate in terms of its interpretation for our observed expression. But as what I just mentioned, it's surely not 100% accurate. But that's the thing we are working on, and probably we could work. I'm not sure whether it's your question, but yeah, probably we can talk some later.
So,
right?
so maybe just quickly, my question was, all these edges between these nodes, you derive from priors. Because if you show PPI, CAC, and so on, what is your experience incorporating these priors? Are some of them better than others in your modeling approach? Or did you combine all of these together?
Yeah, frankly speaking, we don't see big differences across the different knowledge bases. Actually, we have incorporate different databases, like the CAG you just mentioned. But with the real power of our model is the combination of the knowledge part and the data part. So we actually have to combine those two together. So far, we believe the current knowledge base are good enough for either database. So far, yeah.
Thank you.
Thank you. Yeah.
Hello. Hi.
Yeah.
Great work. I have a question about the decipher method that you use to combine the single cell and spatial data. So in that, you learn the share weight between the single cell modality and spatial modality. Did you try to look at the cell-to-cell interaction to see whether the spatial modality can inform the single cell and the other way around?
Yeah, that's sort of the decipher, right?
Yeah.
Yeah. So basically, as you can see, that's something rather close to the core idea of our clue. Because surely there is a lot of interactions between the inner and outer walls of the cell, but we can still separate because we actually can model the outer world or the cellular environment as a topological connected graph. So we can actually evaluate the contribution of each part, the inner and outer, in terms of the expression changes. For just like Sarah mentioned, it works for the clue because the relationship, the crossing, helps to improve the interpretation ability, the explanation ability, and also these things happened here. Actually, I believe Chen Rui has a poster. So you actually have the poster posted, right? Yeah. So we actually have some case studies for doing the the localization related molecules identification based on our disentangling representation. So it works much better than the canonical holistic ones. So I believe that's sort of mathematical things, but I believe that's the separation between the spatial and molecular, the cell identity, has some biological root.
Maybe last question.
Hi, I want to ask a question for glue. So is my understanding correct that in a network inferred by glue, all the edges start from the transcription factor? And if that is true, is that possible to incorporate like a non-transcription factor regulatory pathway in glue?
It's possible for cascade, but it's rather hard for glue because the glue is still correlation-based algorithm, but the cascade, as what I mentioned, is designed for causality.
So for cascade, there's no like pre-assumption of any function for the genes, right?
Actually, we do not incorporate the functional annotation for the data-oriented searching, but we will take those information into account for the later purging. So as you can see from, I believe that's okay, this one. So for the data-oriented searching, we can have a rather good skeleton, but we also need the knowledge purging so that we can reduce the false positive and false negative. So it's again combo effect.
Okay, cool. Looking forward to it. Thanks.
Yeah, thank you.
I might have a last question. For this Swen algorithm, you showed that you can predict the effect of genetic alterations on the gene expression profile. Did you test the limitations kind of about the size of the alteration and the distance from the gene?
Actually, the Swen works for any regulatory changes, including the structure variation and the SNP, and also works for the different part of the genome, right?
Great, super useful. Thank you very much. Thanks again for giving the keynote today.